sir. Am I
Yeah,
audible?
good morning. Can you hear me?
Yes, sir. A very warm welcome, sir,
Thank you. Thank you so much.
on behalf of Biotechnology and also the SRM Institute of Science and Technology, sir. And we'll start the session, sir.
Perfect.
Just
I'm
2
waiting.
minutes. Yeah. Thank you,
No
sir.
problem.
Yeah.
No
Thank you.
problem.
Thank you so much, sir.
Thank you.
தானே இருக்கு.
Thank you for waiting, sir. Honorable dignitaries, learned delegates, respected faculties, and participants. A very good morning to one and all. I'm Aishwarya from Department of Biotechnology, College of Science and Humanities, SRM Institute of Science and Technology.
I'm Smriti from Department of Biotechnology, SRM Institute of Science and Technology. And in the technical team, we have Vijay Raghavan from Department of Biotechnology, SRM Institute of Science and Technology. We feel privileged to welcome you all again for the second day of International Conference on Recent Advances in Biotechnology and Environmental Health Research 2021.
Well, as Albert Einstein said, if we knew what it was we were doing, it would not call me research, would it? It is like an occasion like this, we get opportunities to test knowledge and understanding. We look forward to get an exposure about the best of brain thinks about this very dynamic topics. Let's have a quick recap of the previous session. The first day of the conference was very dynamic and full of insights. We had a great time with honorable dignitaries and 4 keynote speakers who talked about, is insulin a culprit of metabolic derangement causing diseases? The regulation of nuclear transport factor by post-transition modification. Recent progress and perspectives on the role of wind toxin as potential anti-cancer agent. Recent advances on algal biomass utilization as feedstock for biofuel production.
To kickstart today's session, it is with great honor that I may now introduce you to our first keynote speaker of the day, Dr. Sahaya Praveen, to present a talk on SARS-CoV-2 wastewater surveillance with droplet digital PCR. Before that, I would like Dr. Priya to share a few words about Sir. Ma'am, over to you.
Good morning to one and all present here. Nothing is impossible. The word says, I am possible. It's an immense pleasure for me to introduce Dr. Sahaya Praveen, Laboratory Manager for Specialized Medical Solutions, taking care of BioRad Life Science Division as well as Illumina in Qatar. He got his doctorate degree in biotechnology from Manonmanim Sundaranar University, Trichy in the year 2006 and is postgraduate from Bharatidasan University, Trichy in the year 2001. His journey of work started in the year 2004 as a lecturer, Department of Biotechnology, Satyabama University, Chennai. Our dignitary has finished his postdoctoral fellowship in the year 2007 entitled Pharmacognostic and Pharmacogenomic Studies in Ethnic Quartary Populations. He has remarkable experience in applications of genomics and proteomics, divisions of biology, and has ability to explain complex technical and conceptual information in simple language in order to facilitate user learning. Our honorable guest has published 16 research publications and hands-on experience in the fields of molecular biology, microbiology, cell culture, enzymology, bioinformatics, and many statistical tools. Welcome once again, sir. Thank you.
Thank you. Thank you so much for the nice introduction. Good morning, one and all. My name is Dr. Sahai Praveen. I am from a company called Specialized Medical Solution. We are based in Qatar. We have been giving research solutions here in Qatar in the field of both genomics and proteomics for the past 9 years. Allow me to share my screen.
Yes, sir. You can share your screen now, sir.
Is it visible?
No, sir.
One moment.
Yes, sir. Now it's visible, sir.
All right. So today's topic, what we are going to deal with is the SARS-CoV-2 wastewater surveillance with droplet digital PCR. Can you able to see my cursor as well, right?
Yes, sir.
Perfect. So today's agenda is we would like to give the introduction of the droplet digital PCR and the wastewater based epidemiology. So under the wastewater based epidemiology, we'll be dealing with story of the SARS-CoV-2 wastewater surveillance, recent publications highlighting the field of wastewater based epidemiology and the future directions. So before entering into the wastewater-based epidemiology, I want to give the introduction to the droplet digital PCR. So, you all know that PCR has been invented by Carey Mullis in 1983. And as of now, we have 3 generations of PCR. The first generation PCR is the traditional conventional PCR, which is a qualitative one. The polymerase chain reaction, we have 3 steps: denaturation, annealing, and extension. And you will be having several cycles and in order to amplify your target DNA. So at that particular era of time, this particular technology is very, very important. Why? Because this is the only tool available at those time to pull out your target gene or identification of your target gene. But it was qualitative. But the one and only issue with this particular PCR is you have to really wait until your entire cycles to be get completed to check whether your amplification is present or not. Meaning after 30 or 40 cycles, you have to take out your samples, you have to run your agarose gel and to find out whether your target gene is present or not. Whereas in the second generation of the PCR, which is the real-time PCR, we have fluorescent based, and in the terminology real time itself, in the real time, in each and every cycle, you can able to identify the amplification has been performing or not. So it is a relative quantification with your housekeeping gene in order to find your upregulation or downregulation of your particular gene. This is the second generation of PCR, which came on 1993. And the third generation of PCR, which is the droplet digital PCR, which we are going to focus more on today's topic. This is the third generation, and it is an absolute quantification. So it's more or less like the normal PCR, but one and only thing is we are partitioning the samples into 20,000 small droplets. And here, the 20,000 small droplets, each and every droplet will be acting as a reactor, as a PCR reactor, and you will be performing your presence or absence of gene. So the important point here is each and every droplet, if you have the fluorescent, meaning that you have your target gene, and if you don't have the fluorescent, meaning that you don't have the target gene, is a direct counting, and it doesn't need to have any standard curve like that of your real-time PCR. Whereas we rely on the standard curve here in real-time PCR. For the droplet digital PCR, since it's an absolute quantification, we don't need to have a standard curve, and it is very less sensitive to the PCR efficiency, meaning it is more efficient, and it is more tolerant to the PCR inhibitor. This is a very important point. You know very well that as far as the PCR is concerned, there are some certain inhibitors present in the sample itself, and it will inhibit your PCR reaction. Here, this third generation PCR is more robust, and even if you have a PCR inhibitor, that is not going to affect your PCR reaction. That's what they mean to say. Okay, so this is a pictorial representation of this one. You have the samples, and this sample is being partitioned into several droplets. So you just imagine this is the droplet which consists of your target DNA, and these are the droplets. They may have a DNA, it is not the target DNA, let's imagine. And you do the amplification. So amplification is done like a real time, meaning you'll be having fluorescent probes. And then once the amplification has been done, you can go for the reading. So you can able to see wherever your target DNA is there, it has been fluorescent, and wherever the droplet you doesn't have the target DNA, it will not be having any fluorescence. It's a very simple reading. Whatever the fluorescent is positive, meaning you have your target DNA, and whatever the non-fluorescent droplet, which is shown as a black, is the negative, meaning it doesn't have is plus or 1 or 0. 0 means you don't have the target, and 1 you have the target. So this is the pictorial representation of your droplet digital PCR. And this particular thing will explain you the power of the partitioning. You just imagine you have the sample here, and here you have the sample, but you are partitioned into 20,000 droplets here. Here you have a case. In this sample, you have 40,000 wild type. Out of this 40,000 wild type, you have 40 mutant molecule. And our aim is to find the mutant molecule. So since it is a whole mass of sample, you can able to see 40,000 wild type molecule and the 40 mutant molecule. The mutant abundance here is 0.1%. Meaning the primer and probe you're adding inside, it has to go and bind to your mutant molecule, but the probability of meeting your mutant molecule having the huge background of wild type is 0.1%. But in the same case, this scenario, if you're doing the droplet digital PCR, making into 20,000 partition of the same sample, you can able to see this is one droplet. In the single droplet, you have 2 wild type and 1 mutant. So this, as I mentioned, the entire droplet will act as a PCR reactor. It has the primer, probe, DNTPs, buffer, everything. So here, the meeting of the primer and probe is much more easier when compared to the whole mass. So when you go for this logarithmic calculation, it is 33% mutant abundance in a single reactor. So the power of this partitioning is you will not even lose a single mutant gene which is present in the huge background. So that is the power of partitioning. You can see that 40 droplets you can find with the mutant, and the 19,960 which we made 20,000 will not be having anything. So it's a very powerful tool, so to speak. So this is the experimental workflow of the droplet digital PCR. So we have to do the sample preparation. The sample preparation is very simple. You'll be having two channels. In one channel, we have to add it's an oil from the biorad, and there is one more sample channel we have to add your sample. So each and it will go and come and meet. It will be get encapsulated here like this. So this particular sample preparation can be done by two methods. One is an automated method and other one is a manual method. Once the droplet has been generated, then you have to do a normal thermocycler. But this droplet, as I mentioned, each and every droplet will act as a PCR reactor. It has the primer, probe, everything. And if you have your target gene, you will be having the fluorescence. If you don't have the target gene, you don't have the fluorescence. Once the PCR, the amplification is being performed, you have to read it. So each and every droplet will be passed on a small microfluidic channel. And wherever you have your fluorescence, it will be counted as 1. And wherever there is no fluorescence, it is counted as 0. So the end result will be like this. So you can see all the blue dots are nothing but each and every dot represent one droplet and all were positive. And all the gray dots you can able to see or each and every dot represent a droplet which doesn't have your target DNA. And this pink line is your threshold line. So you can able to see it's well separated and there is no cross contamination or something. So this is the same thing, but it is a green droplet because we know that we have FAM and HEX. If you want to have a duplex, let's say 2 gene you have to find, you can do in the same reaction. So one gene will be colored with the blue, another gene will be colored with the green. So you can do the duplex assay as well. And in the 2-dimensional pictorial representation, you can able to see these were the droplet which consists the gene 1. These were the droplet which consists of the gene 2. And these were the droplets which contain both the gene 1 and gene 2. And these are the droplets which doesn't contain any of the gene. So this is a presentation, the paper published here, which has been mentioning the power of the ddPCR with related to the qPCR. So as I mentioned before, we have classical traditional PCR, real-time PCR, and digital PCR. I would say that the real-time PCR is no way inferior for the whatever the test we have been conducting. But digital PCR, when compared with real-time PCR, it's much more superior, meaning the sensitivity is much more. So this particular paper has given you how powerful the droplet digital PCR is. If you could able to see this particular plot, this is an amplification plot. It's a real-time PCR amplification plot. What they have done is, in different concentration of the DNA, they have run the real-time PCR. We all know these are the CT value cycles. So if the amplification gets started in the early cycle, meaning that they have more number of DNA. If the amplification curve start in the late cycle, meaning that you have less number of DNA. If you could able to see here, normally in real time, we run duplicates in order to find the reproducibility. In all the things, they have run the duplicates. If you could able to see the early CTs, there is no much difference. It's a single line. Whereas if you could able to see the late CTs, you can able to see the difference because you can, and you in the fifth line, you can able to see it's well differentiated, even it is 1 CT difference. Whereas in the higher concentration, it is not like that. So the real-time PCR is very good and it is performing very well in the higher concentration. When it comes to the lower concentration, the reproducibility is a question mark. That is what I wanted to show. And this can be rectified by the droplet digital PCR. So here you can able to see the droplet digital PCR amplified plot where you have all the dots here, which is the amplified product. You have your target, and these are the negative droplets which doesn't have the product, and you have the threshold. So you can very well see it is well differentiated. And even if you run duplicate or triplicate, there will not be any of the issue. So this particular paper is been mentioning that droplet digital PCR is working very well in the lower concentration of your template. And moreover, it is an absolute quantification. That is also a plus point, and it doesn't need a standard curve. In the qPCR, what we do, if you want to do a upregulation or down regulation of your target gene, we have to take a housekeeping gene as a reference to normalize your target gene. Whereas here, if you really want to work with your target gene, we don't need to run any of the standard curve, and you can directly go for the absolute quantification. And as I mentioned, it has a high efficiency, so less affected by the poor efficiency, and it has a more sensitivity, even in the lower concentration, and reproducibility. Whenever you are running duplicates or triplicates, the result is going to be the same. So with, as I mentioned, the droplet digital PCR is a superior PCR technology. We have lots and lots of publication, nearly around 6,000 publication has came by using this droplet digital PCR. So now we are going for the wastewater-based epidemiology. We recently found that, like whenever you really wanted to test a viral load in a human sample, let's say for an example, hepatitis B or hepatitis C or HIV, normally they retrieve the blood sample and they'll be doing the real-time PCR to understand the viral load, whether the presence or absence first, and if it is being present, how much copies of the virus is being present. So this is how it has been done. And recent after the pandemic, what happens is like now, since it is a COVID is a respiratory tract virus, we are either taking a nasopharyngeal or oropharyngeal, and then we are doing the real-time PCR. Here, this particular paper is a very interesting paper. What they did is they have checking for the SARS-CoV-2 virus, but in all the clinical samples. So let's first thing you can see the branchiovascular lavage. So this particular sample is taken directly from the alveoli. If you could able to see any of the paper, the best sample to be taken to test whether the person is affected by COVID or not, this is the best sample, but it is more invasive. We cannot able to take it that easily. That is the reason we are going for the nasopharyngeal and oropharyngeal. So, but if you could able to see this, in this paper, they have gone for several blood, urine, feces, everything for the same patient. In this, we found that in the feces, in the stools, they can also able to find the virus. And more importantly, if you could able to see the cycle threshold, that is the CT value on the real-time PCR for that particular individual, in the pharyngeal lavage, this particular size CT is 31.1. Whereas in the feces as well, that is the stools as well, you can able to find it is 31.4. So the CT value is almost picking to a sample which has been recommended for the upper respiratory tract. So meaning that the stools, the feces consist of or it has been projecting the same viral load which the human has been having in, which has been indicating in other specimen site. And in the right side, you can able to see the table. There are lots and lots of publication. They have mentioned that viral load in stool is also been there. So this particular paper has sparked the idea, if in the feces we are having the viral infection, why we can't go for the wastewater identification of your COVID-19. So this is a paper which has been mentioning the monitoring of the waterborne RNA viruses before pandemic. So before pandemic also, we have lots and lots of paper came with the help of the wastewater treatment samples. They have been doing the RNA studies or the presence of the RNA studies. So this is it's a paper which they have given that RNA viruses can be able to find in the wastewater. Here you can able to see this particular diagram, and the black one or nothing but these are the droplet digital PCR, and this gray one or nothing but the real-time PCR. So they have undergone this particular study. Here you can very well able to see that since I mentioned the black one or the droplet digital PCR, in the lower concentration also it has been picking. Whereas the gray one, which as I mentioned, the real-time PCR, there is no lower concentration. The lower concentration is not been picking. So the threshold you can able to increase or decrease. That is we have the choice. But the thing is, like, we are losing the lower concentration of the RNA in the real-time PCR. So in this paper, they are mentioning The droplet digital PCR is much more superior than the real-time PCR in identifying the RNA viruses in the waterborne, because they are lower sensitivity because the limit of detection is more, and high variability between measurements is also being measured. And more importantly, the qPCR is more prone to inhibitors, which is not being the case of the digital PCR. And there are lots and lots of other publications also been published where the waterborne RNA viruses can be detected by digital PCR. This is another interesting paper. This is from the Netherlands. So what they did is they have identified 3 genes, N1, this is a N1 gene, this is N2 gene, and N3 gene, and as well as the E gene as well. So this is they have used a droplet digital PCR. Here, what they have correlated is, you know that each and every country in the Ministry of Public Health, every day they'll be having the COVID case study. So in parallel, what they did, they have also gone for the wastewater droplet digital PCR, and they found that the droplet digital PCR COVID presence data is much more higher when compared with the MOPH, that is the Ministry of Public Health published data. Meaning, see, normally whenever the patient is coming, the patient, whoever having the symptom is coming for testing, and whoever being tested, the result is coming whether it is the patient is being positive with COVID or not. Whereas the wastewater treatment, if there is a person who are asymptomatic or pre-symptomatic also, even they are shedding the virus in the stools, that is also being counted. So the underreporting can be very well checked by the wastewater treatment when compared to the MOPH. So if you, for an example, in US or Australia, or even in Europe, you have small counties, small area where you have a wastewater treatment. Like, let's say you have 100 families living together. You can take the wastewater and you can find whether the presence of COVID is there or not. Meaning, if there is no, means that the entire population is free of COVID. So this is it's a surveillance check that we need to do it in the coming years, and it is very important. So this particular digital PCR will fetch you that particular luxury to identify the surveillance. So here the under testing has been reported, and moreover the increase in clinical cases is correlated with the wastewater finding is also been there in this particular paper. And this is a paper which is much more interested, and this is a SARS-CoV variant paper. But here, this paper has illustrated the detection of the variant of the virus. Normally, we are checking the presence or absence of the COVID, but in this paper, we are checking the presence of the absence of the variant. Let's say, you know, that there are alpha, beta, gamma, delta variant, and this N501 is the UK variant. Now it has been designated as an alpha variant. So this particular variant is there or not can also be done with the droplet digital PCR. So how they are doing is we will be designing the primer and probe accordingly, which will go and bind to this particular mutation. And if it is being the presence of that particular alpha variant is being there, it will be get illuminated, and we can be able to identify this strain is there or not. So this is the first paper came to identify the variant. And you all know, if you really wanted to identify a variant, we have to do the sequencing in the normal scenario. So even if you have your real-time PCR, it will let you know whether the presence or absence of the virus. But if you really wanted to know whether the virus which is being present is an alpha variant or beta or gamma or delta, you have to do the sequence. But this digital PCR, with the power of your primer and probe, we can also be able to identify the UK strain, which they have published as a paper. This you can able to see the pictorial representation. In the left corner, they have done in 2 different cities in Amsterdam and Utrecht, and you can able to see the blue one. The blue one is nothing but the UK variant. And the orange one is nothing but the data published from the Ministry of Public Health of both the things. So you can able to see it is correlating. These are the different months they have taken. So it has been correlating with the wastewater identification along with the Ministry of Public Health, which they published. It is in Amsterdam. The same way in the URIST as well, it has been correlating with the same way. Meaning that the tool what we have been using, not only the presence or absence of your virus, but also the variant of the virus is also been very sensitive and it is very precise, where we are exactly or nearly matching with the Ministry of Public Health report here. This is another paper. It's from Southeastern Virginia. Here, it's very interesting study. If they have done the wastewater treatment, wastewater sample digital PCR along with the MOPH, the Ministry of Public Health reported, you can able to see it's way ahead. This is the line which shows you the wastewater treatment COVID detection, and this is the line which shows you the actual testing, clinical sample testing thing. You can able to see it has a very big difference, and they found that it is underreported. You can able to see they are also been looking for 3 genes, N1, N2, and N3, and these are the diagrammatic representation that each and every date they have reported, and they have plotted here, which when they have been very much surprised to see it was underreported when compared with the MOPH. So meaning that there are a lot of people. This is surveillance. So we have to come to a conclusion that there are a lot of people, they are asymptomatic and pre-symptomatic. That is the reason we can see the deviations here. So, and the droplet digital PCR is also being applied in several other areas of environmental testing. Let's say a very important thing is your aircraft, because most of the infection is through coming in the aircraft. That can be taken into surveillance. You can take the sanitation system and you can do the droplet digital PCR in order to understand whether the aircraft is fully COVID free. And the right side, there's a Nature, they have published the paper. They have mentioned that in the Wuhan hospitals, so many viral load is being there in the aerosol, and it is more importantly in the toilets used by the patients. So in order to give that particular environment as a viral free, or to monitor the particular environment as a viral free, we have to capture the aerosols, and we have to do the droplet digital PCR, which will fetch you the results, whether it is free from COVID or not. This is a community, there are a lot of community has come. This is a COVID-19 poops dashboard. This is a dashboard is meant for the COVID detection from the stools or the wastewater. So there is a website you can able to see, and you can understand how many countries are doing, how much is the paper has been published, everything is been reported in this community. What are the current challenges and the future directions? So there are certain challenges while we are going for the wastewater epidemiology. Meaning, the first one is shedding probability and magnitude. So we do not know, because it is not any controlled environment. If you're going for the wastewater treatment, we are not sure that wastewater sampling what we have been doing will be concurrent with a sampling we have been doing in another country, because it is highly variable. And that the viruses are RNA, and RNA can also very easily decay in the kinetics of the wastewater. So that is also one of the major challenges we are facing. And the complexity of the wastewater. Sometimes what happens if there is a rainy season, the samples will be more diluted when compared with the sampling what we did in the previous months or previous years. So sampling techniques and storage condition, we need to have a standard for it, how to do the sampling. And once it has been sampled, how to store it. And what is the concentration method we need to follow. And more importantly, what is the extraction method? As I mentioned, if you want to isolate an RNA from a sewage, you need to really have a robust extraction methodology, because the sewage will consist of lots and lots of background in terms of inhibitor. Whatever the method you have been implementing to extract, that should be more sensitive, robust, meaning it will take only the nucleic acid, removing all the remaining efficiency. That has to be done. And the detection method should be, we have to standardize. And more importantly, the population size. We have to normalize it. So we cannot compare with a 100 population city with a 1,000 population city. So how to normalize these things? So these are the challenges in this wastewater sewage methodology. And as I mentioned, this has to be we have to work hand in hand. Molecular biology has to be interrelated with the MOPH people, public health expert, as well as the wastewater infrastructure experts. So they have to work hand in hand in order to figure out how to clarify all these challenges, and then we have to make a standard operating protocol for that. So in summary, as I mentioned, ddPCR is very sensitive when compared with real-time PCR. So improved sensitivity. Even if you have a very low viral load, it can be detected, reducing the false negative results. As we mentioned, as we saw in our earlier slides, it's even it will increase the abundance, and so that you will not lose not even a single virus in this particular technology. And it has the ability to quantitate without need of standard CAD. As I mentioned, we don't need to have any housekeeping gene in order to have a standard relative quantification, because it is accurate and it's an absolute quantification. So it is, results are very easy to interpret. As I mentioned, it's a presence or absence, 0 or 1 in the droplets. And it is unparalleled precision and a very high data reproducibility. And we have nearly 5,900, nearly now 6,000 publications in various fields in liquid biopsy and environmental monitoring, etc. Thank you. I'm happy to take any questions.
Thank you very much, sir, for sharing your deep knowledge with us. Your session was full of enthusiasm and valuable knowledge. Now the session is open for discussion.
Good morning, sir. This is Dr. Mani Mekle from the Department of Biotechnology, sir.
Good morning.
And I have a question regarding this, because whenever anything that we're working on with an RNA, we will always look for the stability of the RNA. Whenever isolating an RNA, immediately we will go for the cDNA conversion for the better storage for the future experiments and all. So for this, when we are using this ddqPCR, any special method of the RNA isolation has been prohibited by this, or the normal way of the RNA that we can go for, sir, for isolation techniques?
Normally now, we are depending on the magic mixtures. For an example, if you take a company called QIAGEN, they are giving RNA extraction, which is meant for wastewater. Okay, so normally the laboratories, they go for the QIAGEN RNA extraction for the wastewater, and they are extracting the RNA, which is more robust and it is free from inhibitors as well.
Okay, sir. Thank you, sir. And also this ddqPCR detection of the mutant, as you said, like the variant identification of for the SARS-CoV, like I need some more information, sir. Can you explain it again? Because it is so interesting, something which we identified, and it can SNP identification, like generally we will go for the microarray and all, right now, sir. So how far it will be helpful when you're going for this ddqPCR to identifying the mutants?
Okay, so like each and every alpha, beta, gamma, and delta, we know that there is a specific deletion or a substitution or some changes, right? So we are going to design the primer and probe in such a way it will go and bind to that particular flanking region, and it will give you, because normally what happens if you're going for a variant, we will not look for one particular gene. We'll be looking for 2 genes to normalize. For an example, I'll give you an example. You have 10 people affected with the COVID. Out of 10 people, we have only 2 people affected with the Delta variant, let's say. Now, you have to first identify the 10 people, along with you have to identify whether it is Delta or not, meaning you have to probe 2 regions, one for, let's say, nucleocapsid and one for that particular deletion. So you'll be having the results of 10 along with 2, meaning it has the 10 people has been there along with your Delta variant. So we have to always correlate or normalize with the normal gene along with your variant of your interest. But the variant, as I mentioned, depending upon that deletion or substitution, you have to design the primer and probe, which will go and fit like your normal SNP.
Yeah, thank you, sir. Thank you so much, sir. Thank you.
Thank you, sir, once again. Moving on next, now let's begin our oral presentation. Participants are requested to acknowledge the attendance.
All right. Before, I just wanted to thank the organizing personalities of this conference. And I would like to thank the convener, Dr. Suresh, as well as the head of the department for giving this opportunity. Thank you. Thank you, one and all, and good luck.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you.
Thank you.
So let's start with the oral presentation attendance. So oral presenter 1, 31, Shanir, Department of Mechanical Engineering, SRM IST Katangutu, Chennai. Oral presenter 32, P. Araga Sarvana Babu, Sagorika Goyal, Vijaya Afrin, M. Sugukumar, Institute of Science and Technology, Avadi, Chennai, Center for Food Technology, Department of Biotechnology, Anna University, Chennai. P. Aragana Saravana Babu, Madhushri Shankara, Shyam Nirmala Nishal, Sai Nandini, Jayanti Parnivelu, Department of Biotechnology, Veltek Ranganajan, Dr. Shakuntala, R&D Research Institute of Science and Technology, Chennai. Harikulasi, Dr. S. Subramaniam, Dr. Shyama Subramaniam, Regenex Super Speciality Laboratories Private Limited. Ram Prakash Balasubramani, Dr. Erin, Laboratory of Cyanobacterial Biotechnology, Department of Biochemistry, Faculty of Science, Chulalongkorn University, Bangkok, Thailand. Dr. M. Rizwan Ahmed, Assistant Professor, SRM IST Ramapuram Campus.
Participants kindly wait for 5 minutes.